let's talk about lipid catabolism. So now we're actually talking about breaking down lipids. We're going to talk mostly about beta oxidation, so the process of breaking down fatty acids. But the way that fatty acids are stored is typically in the form of triacylglycerols. So where you have the fatty acid, that's actually, you have three of them that are hooked up to a glycerol backbone. Remember how we talked about that was a really great way to kind of like store energy in this compact form that wasn't going to hold on to water? But when you want to mobilize those fatty acids, mobilize like fats, the energy from it, well, those triacylglycerols aren't going to be so great. And we're going to need to actually chop the fatty acids off of the backbone, which we can do with a couple different lipases that we'll talk about. I don't need you to worry too much about, or pretty much at all, about this digestion and mobilization of fats. I want you to focus on the beta oxidation. But because this is an important topic medically, so we're talking about things like low density lipoproteins and high density lipoproteins, I want to give you a little exposure to it, as well as provide resources if you would like to learn more. But that being said, here's a big, overly simplified, generalized talk about what happens to those fats and when we eat them, how they get stored and shipped, how we can basically provide fatty acids to tissues throughout the body. All of this is mainly thanks to our liver, our adipocytes, so our fat cells, and our small intestines. Remember how we can make fatty acids and lipids in our liver and in our fat cells, so our adipose tissues. We can also break down those triacylglycerols that we make in our liver and in our adipose cells. Our liver is going to send triacylglycerols through the bloodstream in the form of these kind of lipidy bubbles called lipoproteins. And our adipocytes, so our fat tissues, when they break down lipids, if they want to send them to the body, they send them as fatty acids that are kind of like traveling, piggybacking on proteins like your serum albumin. Because remember, those fatty acids, even though they're not quite as hydrophobic as those triacylglycerols, they're still going to be very hydrophobic. And so they're going to need to ride through the bloodstream. So how is your liver able to send them through the bloodstream? Well, they do them in this lipidy bubbles called these lipoproteins. So these lipoproteins, they're like these little bubbles. Unlike your cell membranes, which have a phospholipid bilayer, your lipoproteins, they have a monolayer. So they just have this one layer of these phospholipids, which is great because, well, now they have these hydrophilic heads that are out facing the watery surface of in your blood. And they have this hydrophobic, these tails inside where they can glob up and hold on to various other hydrophobic things, like more like triacylglycerols, your tags, your cholesterol, and various things. These lipoproteins get their names for the protein, but there's not actually much protein in these things. But each type of lipoprotein has these various apolipoproteins, so these proteins that are embedded in them that are actually going to let them dock onto various cells and things. There's a similar type of molecule called a chylomicron, so a similar sort of like bubbly thing. The chylomicrons are going to be sent out by your small intestine, whereas those lipoproteins are going to be sent out by your liver. So when you ingest a meal that has some fats in it, what happens is that in your small intestine, it's going to package those fats up in these chylomicrons. So think of the lipoprotein, but slightly different. And they're going to be bigger. And these are sent out by your intestines. And then they go and they travel through the blood and they get to your liver. And now your liver's job is to kind of like divvy things up. It's going to, in order to take in those triacylglycerols, well, you can't really take them in as is. And so instead, what's going to happen is it's actually going to chop the fatty acids off of them and then take those in. The way that you kind of chop off the fatty acid tails from extracellular tags, so ones that are kind of like outside of cells in your bloodstream, is that in the walls of your like endothelial cells, so in your kind of the walls of your blood vessels, there's this lipoprotein lipase, and it can chop off the fatty acids so that the fatty acids can come into the cell, whereas the tags could not. So your liver is going to go and it's going to take in the fatty acids, and now it has to decide what to do with it. It can basically take those fatty acids and turn them back into tags in order to store them. It can break them up down for energy in that process of beta oxidation that we'll talk about, or it can ship them to other tissues in need. And so, well, it's going to ship them, actually, it's going to ship the triacylglycerols in those lipoproteins. First, it's going to send out, what it's going to send out directly is this like very low density lipoprotein. And these are kind of these bigger bubbles. And these bigger bubbles, well, they're going to be very low density because, well, they're mostly filled with these triacylglycerols, which if you think about fats and like fat floating and stuff, it's not very dense. What happens though is that other tissues are then going to go and they're going to take in fatty acids. So those lipoprotein lipases are going to break off fatty acids from tags from these lipoproteins, and that's going to allow fatty acids to be distributed to other tissues. As this happens, that lipoprotein is going to shrink in size, and since you're getting rid of the tags, it's going to get denser, and so you end up with intermediate density lipoproteins, and then low density lipoproteins, and then high density lipoproteins. And those high density lipoproteins are going to go like back to your liver and stuff. So high density lipoproteins are typically considered like your good cholesterol, where it's like your liver kind of taking in stuff. And then the low density lipoproteins, that's kind of like the bad cholesterol. That's your liver like sending cholesterol and things to other tissues. Gross oversimplification, but that's kind of the big picture idea in a way. So your liver is basically sending out these tags in these lipoproteins, as well as cholesterol and other lipidy things. Your other tissues then take them in and do with them what they want. One of the main tissues that's taking in these fatty acids are your adipocytes, so your fat cells. Inside of your fat cells, well, they can then go and convert them back to tags in order to store them. Yes, it's weird that you're basically keep chopping them off and putting them back and chopping them off and putting them back. Well, at least the putting them back together, that will happen in your liver and in your adipocytes, where we can actually store these as tags. In your other tissues there, you're typically just going to be wanting to break them down for energy. And we're going to talk much more about that breakdown process. So your liver and your adipocytes can both store fatty acids. They can also break down those fatty acids if they need energy, or in the case of your adipocytes, the way that they actually ship fatty acids ship fatty acids to other tissues is not as tags in lipoproteins, but instead as fatty acids that are being carried on proteins like your serum albumin. So it needs a way to break down those intracellular tags, and so does your liver. Whether it's the tags that they have basically made themselves from scratch, or by piecing back together the fatty acids. In either of these cases, they need to be able to then break down those tags intracellularly. Lipoprotein lipase was extracellular. If we want to break down intracellular things, we're going to need a different lipase. It'll do the same thing, but now we're working intracellularly. And now we're going to talk about hormone-sensitive lipase. Basically, what it does is it's going to, as the name suggests, be responsive to hormones. And the hormones that's going to respond to are like glucagon and adrenaline. So remember that those were kind of telling us either we need to break down energy because there's a stressful situation, we need energy, or we need to like mobilize energy to get things to fuel to other tissues. Both of those were able to kind of do similar things in some cells because both of them were going to activate the same kind of, they're both going to activate the adenylate cyclase, which is going to activate our pKa. So both of them are acting through G protein coupled receptors. that activated adenylate cyclase, generated cyclic AMP, activated pKa. And in this case, what pKa is going to do is it's going to activate protein kinase A, which is going to phosphorylate and activate hormone-sensitive lipase, which is then going to break down those tags into fatty acids. Conversely, well, if we said phosphorylation was going to activate it, dephosphorylation will inactivate it. And so we would want to inactivate it when we have plenty of fuel. We want to be like storing these lipids as tags. Well, then we wouldn't want to break them down. And so insulin is going to say, activate this protein phosphatase, and there it's going to dephosphorylate this so you stop breaking them down. We'll revisit this topic, but it should be reminding you of some of the things about like glycogen metabolism. For now, let's talk about how we actually do that breakdown of the fatty acids, that is the beta oxidation, now that we've talked about kind of like how we could free up those fatty acids. And again, don't worry about the details of freeing up the fatty acids. Let's focus on the beta oxidation. Fatty acid breakdown or catabolism happens in the mitochondria, and this is happening through a process called beta oxidation. It's similar to the reverse of what we were looking at before, in the case that we are breaking down things to, acetyl groups that are 2 carbons at a time. But note here, we're not having a malonyl-CoA intermediate. So when we're building up a malonyl-CoA, that's really indicating that we're synthesizing lipids. Don't worry about the details of these reactions, but basically before we had a bunch of reductions, now we have a bunch of oxidations. How this is going to happen is basically we're going to be doing these oxidations using both FAD and NAD+ to give us FADH2 and NADH. Remember that this is going to be happening in the mitochondria, so that NADH is already going to be where it needs to be to enter the TCA, so it can enter and get the full ATP yield at complex 1. The FADH2, well, here it's going to contribute to our Q pool, but it's not going to pump proteins from that first step. So you're going to get less energy from that FADH2 than from the NADH, but that's just a technical detail. And you're still going to get a lot of energy from fatty acid breakdown. How this breakdown goes down is that basically you're going to do this first oxidation to generate this alkene, this carbon-carbon double bonds. Then what's going to happen is you're going to hydrate that. You're going to add water across the bond. You're going to then oxidize that alcohol group to a ketone. And then you're going to cleave it off. And the way that cleave off the acetyl group, the way that you cleave off the acetyl group is going to be by using a CoA. Remember that our sulfur groups are going to be swappable. So basically we have this, this fatty acid. It's actually in the form of this fatty acid CoA, this acyl CoA, which I'll discuss in a minute. And now what you're going to do is you're going to take another CoA. So when you chop it off, it's kind of like you're chopping off part, but then having your scissors stuck to where you cut. So this sulfur linkage basically is going to come and it's going to attack this carbonyl carbon. You're going to get a bond to that carbonyl carbon, and then you're going to get this end group is going to get cut off. When we were building before, we were building from this from the end that was attached to the protein, to the ACP. And here we're going to be removing from that same end, but here we're attached to CoA. And we're going to be cutting off these CoA pieces one at a time. And remember, each of these CoAs is 2 carbons. So what's going to happen is that we're going to be, if we have an even number of fatty acid, we're eventually going to break it all off into acetyl-CoA. If we have a odd number of fatty acids, which are rarer, but they do occur, then what's going to happen is you'll end up with a 3-carbon compound. So the 3-carbon acyl-CoA that you get is called propionyl-CoA. Propionyl-CoA then gets converted through a couple of different reactions into something that we are definitely familiar with, succinyl-CoA. And where does succinyl-CoA come in? Well, basically, it's one of the components of the citric acid cycle. So odd-chain fatty acids can actually enter the citric acid cycle. The end product of it can enter as the succinyl-CoA. So we get succinyl-CoA from that propionyl-CoA by basically carboxylating it, shifting things around. We get that succinyl-CoA, and the succinyl-CoA can then enter the TCA. This has the consequence of those odd-chain fatty acids actually being glucogenic. That is, we can use them to synthesize on net glucose. This is because it's an anaplerotic reaction, replenishing something from the citric acid cycle, so that we can basically then take out malate to convert to oxaloacetate to go and make glucose from. However, with even chain fatty acids here, we just break them down to acetyl-CoA, which was not in our TCA. So we only can enter the TCA if we have oxaloacetate present. But if we have odd chain fatty acids, we get at least 3 carbons that are going to then join with the CO2 to make 4 carbons that we can then enter into this pathway and keep the cycle going, even if we take malate out in order to go make glucose. Hence, we can say that odd chain fatty acids are glucogenic, whereas the even chains are not. We can also handle if we have saturated fatty acids or unsaturated fatty acids, that is. And so remember that our natural unsaturated fatty acids are going to be cis double bonds. And in the form, and what we're dealing with here is actually going to be a trans double bond that we add in order to break things. So those cis bonds will get converted into trans bonds, and then they can be cut down that way. But you're going to get a little less energy because you don't have that initial oxidation in order to make the bonds. But this is how we're going to be breaking down the fatty acids, and that's going to be occurring in the mitochondrial matrix. But there's a problem, because that fatty acids can't get carried in as in the form of the acyl-CoAs. So in our cytosol, basically what's going to happen is that we can have these fatty acids attached to to a CoA group. And so when you do this, it's done by this like fatty acyl-CoA synthetase. And it's going to take the fatty acid and it's going to put it onto acetyl-CoA. It's going to put it onto CoA. It's going to take ATP to do this because we're forming, remember, this higher energy bond. And so now we have this fatty acyl-CoA, but it can't, this was in the cytosol, and it needs to get into the mitochondrial matrix. We can't take it in as is, and so instead we're basically going to use this one higher energy bond and swap it for another higher energy bond, but this case on the carnitine. So carnitine is going to be another one of these small molecules, and it's going to have a group that can attach to this, to where the CoA was attached in a swappable manner. This will give you a fatty acylcarnitine, and then it can get transferred through this translocase into the mitochondrial matrix. Here, you're going to basically have another carnitine acyltransferase, which was the thing that is going to transfer it onto carnitine. Now you're going to transfer it off of carnitine and onto the CoA. This will give you a fatty acyl-CoA, and that can then be broken down, and then those acetyl-CoA pieces can enter the tricarboxylic acid cycle. So what happens is you have this fatty acid in the cytosol. Fatty acyl-CoA synthetase is going to hook that fatty acid onto a CoA handle. It's going to then swap that CoA handle onto from the CoA onto carnitine. This is done with this enzyme carnitine acyltransferase, or CAT1. Sometimes this enzyme is also called CPT1. So basically the CAT1 would stand for carnitine acyltransferase. But remember, our default fatty acid was our palmitine. So we have palmitoyl transferase, carnitine palmitoyl transferase. So you also see it called CPT1. In terms of the 1 and the 2, well, basically, because you need one version on the outer mitochondrial matrix and one version on the inner mitochondrial matrix. So CAT1 is on this outer, and then CAT2 is on the inner. And we'll discuss in a minute, these are key regulatory points. Once you've swapped your fatty acid onto carnitine, you get a fatty acylcarnitine, which can then go through the translocase located in this inner mitochondrial matrix, give you that fatty acylcarnitine in the mitochondrial matrix. Now we want to get the carnitine back. The carnitine is basically going to, the fatty acid is going to be swapped back onto our CoA to give us a fatty acyl-CoA. And then this carnitine is going to be sent back through so it can pick up another fatty acid. To review, the breakdown of fatty acids happens in the mitochondria in a process called beta-oxidation. We break off 2 carbons at a time, but we're doing it so from the end that's kind of hooked up to the carboxylic acid, which in that point won't actually be a carboxylic acid. Instead, it'll be attached to a CoA group. But we're kind of going to be cutting off from the same end as which we added on in the beginning. And that's kind of going to be the opposite end of the end that you would think. So it's not the free end, it's the end that's actually attached to the CoA. But when we think about a fatty acid, that actually doesn't have a CoA. And so the first step we need to do if we want to break down a fatty acid is to actually link it onto a CoA. This happens through this fatty acyl-CoA synthetase or ligase, this ACS. This is going to link on this CoA. This CoA has several purposes, one of which is going to help you get it into the mitochondria through some sneaky swapping, and it's going to introduce a linkage point at which you can swap things on and off. So remember that CoA, the linkage happens through a sulfur, and sulfur linkages are going to be easier to make and easier to break. Because that's kind of more of a high energy bond, meaning that the breakdown products are going to be more energetically favorable than the formed product, than the initial reactant, than the thing with the CoA bond. That means that you're going to basically be able to make these reactions happen more favorably, but you have to invest energy in order to do so. You have to invest the equivalent of 2 ATPs because you use an ATP, but you go to AMP. This gives you your fatty acyl-CoA, which then you sneak into the mitochondria. I say sneak in because, as we saw when we talked about the citric acid cycle, things with CoA can't come in or out. So instead, we swap the CoA for a carnitine to get us our fatty acid acyl-carnitine, and that's going to enter the mitochondrial matrix via carnitine acetylcarnitine translocase. You basically swap it for a carnitine. Once you're in the mitochondria, you swap it back for a CoA and return the carnitine. And that was what we called our carnitine shuttle. In a little more detail, so we have our fatty acid, and now we want to designate it for breakdown. We do this by attaching a CoA, which is done by this fatty acyl-CoA synthetase, or your ACS. Now what happens is you have a fatty acyl-CoA. This fatty acyl-CoA, however, can't get into the mitochondria, so it has to be transformed into a fatty acyl-carnitine. This happens with the help of this carnitine acyltransferase 1, which is your outer mitochondrial membrane. Now what happens is you get this fatty acylcarnitine. Then this fatty acylcarnitine can now go in through this carnitine acylcarnitine translocase that's located in your inner mitochondrial matrix membrane. And so that fatty acylcarnitine can actually get into your mitochondria. But beta oxidation is going to break down acetyl fatty acyl-CoAs. And so basically, we need to now make this fatty acyl-carnitine back into a fatty acyl-CoA. So we reverse the process that we did above. We swap the carnitine back for a CoA using carnitine acyltransferase 2. So the version that is in your inner mitochondrial matrix membrane. This CAT1 is going to be a key point of regulation because it's how you get these fatty acids into your mitochondria. Okay, and it's going to be inhibited by melanoid-CoA because if we're building fatty acids, we don't want to be making them. Once we bring in these fatty acids, well, now we're going to break them off, and each cycle we're going to break off 2 carbons as an acetyl-CoA unit, which can enter this TCA. And remember, we're in the mitochondria now, so those can just directly enter the citric acid cycle. During the process of breaking it down, not only are you generating acetyl-CoA that can enter the TCA, but you're also generating FADH2 and NADH. The actual beta oxidation part, well, it's happening in an oxidative fashion, hence the generation of these FADH2 and the NADH. And if we think about why, well, we said that fats were good sources of energy because they were highly reduced. They had lots of potential to be oxidized. When we oxidize, we make NADH, we make FADH2, and those can be used to generate energy. But in order to actually get that energy, we need to do that oxidation. And so that happens in this process of beta oxidation. From one molecule of palmitoyl-CoA, you could actually generate a lot of energy. So from the breakdown, we are getting the 7 FADH2 and the 7 NADH, and then the 8 acetyl-CoA. And then we're taking that through the TCA. We're getting 8 GTP, 8 FADH2, 24 NADH, and then all of that could go through the electron transport chain. So lots of energy. And this was just coming from a single fatty acid. And remember that often that's going to be coming from something like a triacylglyceride. And so you're going to get the glycerol as well as all those fatty acid tails. And so fatty acids really do provide a large source of energy because they have all that oxidative potential. Like most things in biochemistry, of course, there are exceptions. Fatty acyl-CoAs that are shorter than 12 carbons actually don't need that whole shuttle process. So see, look here, we actually have a simplification. Basically, they can just pass through the membrane unassisted. We're typically going to consider, though, that we're working with longer fatty acids, but not super duper long fatty acids. In fact, if we have fatty acids that have 22 carbons or more, we call these very long chain fatty acids, or VLCFA. And they're actually broken down in specialized organelles, so not in your mitochondria, at least not first. They're broken down in organelles called peroxisomes, and a different process that is actually going to use hydrogen peroxide. So basically, that's going to be really reactive, and so you want to keep it in these specialized little bubbles inside of your cell, these little organelles, these membrane-bound organelles. I say like initially broken down because they're broken down in there into an 8-carbon fatty acyl-CoA, and then that gets broken down further in the mitochondria. So that one is a little more complicated. And there are disorders in peroxisomes that can actually cause, like genetic disorders that can cause various diseases. Like adrenoleukodystrophy, which if you've seen like the movie Lorenzo's Oil, that was dealing with that. So let's talk a little bit about regulation. Fatty acid synthesis and breakdown are going to be inversely regulated. So if we're making lipids, well, when do we want to make lipids? Well, we want to make lipids if we have plenty of energy and more than we can handle right now. Let's store some. If we're wanting to store lipids, we're not going to be wanting to break down lipids at the same time. Similarly, how we wouldn't want to be doing glycolysis and gluconeogenesis at the same time and breaking down the glucose as we're making it. We don't want to be breaking down our fats as we're making it. And so we want to have the products of this fat making be inhibiting the enzymes that break down fats. The easiest way to inhibit the fats from being broken down, the most efficient way, is to keep them from getting into the mitochondrial matrix, which is where that beta-oxidation was taking place. So remember how milano-CoA, that 3-carbon intermediate we got from carboxylating acetyl-CoA, that was going to be produced during our lipid synthesis. We can have milano-CoA negatively inhibit our CAT1 or CPT1, this thing that was going to take that fatty acyl-CoA and basically hook it on to carnitine to get it into the mitochondria. Rather than taking these groups into the mitochondria, we want to stop that, and so milano-CoA is going to inhibit CPT1. Another key point of regulation is this ACC, or this acetyl-CoA carboxylase. That was how we actually produced the milano-CoA. When would we want to stop this from happening? Well, we would want to stop this from happening if we were building up a lot of fatty acids. If we're building up a lot of fatty acids, maybe we need to slow things down. Use that acetyl-CoA in other ways, maybe. And so we have these fatty acids, so like our palmitoyl-CoA can actually inhibit this ACC. When would you want to stimulate the ACC? Well, maybe you want to stimulate it if you have a lot of citrate. So if you have more citrate than, say, your tricarboxylic acid cycle can handle, or basically you have citrates and you don't need as much energy, what you want to do is basically you want to send the message down the line, hey, get ready for us. So the citrate is going to positively regulate the ACC. So when we have a lot of citrate, that's going to stimulate the generation of these fatty acids. Remember, too, that citrate was a negative regulator of glycolysis by inhibiting phosphofructokinase 1, PFK1, which was that key point of dedication into the pathway of glycolysis. It makes sense that we're kind of reciprocally regulating fatty acid synthesis and glycolysis because, well, if we have a lot of energy and we're wanting to store it as fats, we're not going to be wanting to break down sugar for energy. So if we have a lot of citrate, let's stimulate our lipid synthesis and let's inhibit glycolysis. We also saw how long-chain fatty acids, such as our palmitoyl, such as that palmitate we made, that can actually go and inhibit pyruvate kinase. So pyruvate kinase, remember, this was taking our phosphoenol pyruvate to pyruvate in the process of glycolysis. And so basically, we would want to inhibit it. So we're inhibiting glycolysis in this way, promoting instead the change of pyruvate into that oxaloacetate, and that we can then take through the citric acid cycle in order to get our acetyl-CoA that then we could use to make these fats. So we see coordinated regulation between the different pathways. Another point of regulation is they're like the lipases that are actually going to cut down the tags into fatty acids. And so this can be regulated by things like hormones, various signaling pathways. And so this is all going to be tightly regulated. The fuel from fatty acids, remember, we can break things down to acetyl-CoA, but we couldn't transport the acetyl-CoA that way through the blood. And so instead, what we're going to want to do is we're going to want to convert that acetyl-CoA into things that we can transport through the blood. Especially, this is important for your brain. Your brain's really picky and it won't break down fat for energy. And so if your brain runs out of its preferred food source, it's glucose, well, then your liver needs to send it energy, and it does this in the form of these ketone bodies. These ketone bodies come from acetyl-CoA, but they're not acetyl-CoA. Instead, we have acetoacetate and beta-hydroxybutyrate, as well as acetone, which is just a non-enzymatic byproduct, which isn't the one that you want. Instead, the main form that we have as our carrier is going to be this beta-hydroxybutyrate. It's not technically a ketone body. It's got a carboxylic acid group rather than a ketone, but it can get converted into acetoacetate, which is a ketone, and that can be used to generate this energy. Basically, you can get two acetyl-CoAs hooked together through a couple of different enzymes to give you acetoacetate, which can then get converted into beta-hydroxybutyrate, which is just the reduced form of it. So acetoacetate, it can spontaneously decarboxylate to give you acetone, which isn't what we want. And that's because it has this beta-keto acid. Beta-hydroxybutyrate is a reduced form of this, so we reduce this to an alcohol. We don't have that issue. This is going to be more stable, and we can transport it through the bloodstream. One medical connection notes, this you might hear about this HMG-CoA synthase. There's some intermediates before you get to acetoacetate, but this HMG-CoA synthase is the one that does this initial step. And it's also, this is going to be important for making steroids and making your cholesterol. And so HMG-CoA synthase is actually a target of statin medications for controlling blood cholesterol. But it's also an important step here for producing these ketone bodies. These ketone bodies can then be transported from the liver to tissues like the brain in order for the brain to then use them, assuming that it has F-tricarboxylic acid component still in order to do so. Another medical note is acetone, this spontaneous byproduct that we don't want. Basically, it can form if you have a lot of these ketone bodies building up because there's not enough of the components of this TCA cycle for that acetyl-CoA to enter and be broken down to be oxidized fully. These ketone bodies can build up in the bloodstream. It's just a condition called ketosis. Sometimes you'll see like ketoacidosis. What happens is because if we look at the acetoacetate and the beta-hydroxybutyrate, those have carboxylic acids. They're going to acidify the blood, leading to this condition of where you have acidified blood caused by these ketones, which is ketoacidosis. You see that sometimes with diabetes that's uncontrolled. And you'll also see that their breath is going to smell like nail polish remover or floral scent type, weird fruity scent. That's from the acetone, that bad product that can build up if we can't be handling the things great. Because remember, with diabetes, what's happening is the cells aren't taking up enough glucose. The blood sugar can get high, but also you don't have enough energy inside of the cells. So the cells are starving, even though there's going to be plenty of glucose around, it just can't get in. And the glucose was being broken down in order to give us our tricarboxylic acid components that we could then use to break down, to oxidize things fully, to give us energy. If we aren't able to go through the TCA, well, then we're going to basically have this acetyl-CoA build up. If that acetyl-CoA builds up, well, now it's going to go do alternative things. We could make fatty acids with it, but that wouldn't be very helpful for us here because, well, what we need is energy. We're not trying to store fatty acids. And so instead, we get this problem, we get this build up of these ketone bodies. And so that's often a diagnostic sign of uncontrolled diabetes, is having that like acetone breath. Fats aren't the only way that you can get these ketone bodies. They can also be produced by some of the amino acids. Key takeaways: our fatty acid synthesis is going to be happening in the cytosol of our liver and of our fat cells. Our fatty acids are then going to basically be stored in the adipocyte, so in your fat cells and your liver cells, and then they are kind of broken up by hormone-sensitive lipases in response to energy signaling. So if we have glucagon or epinephrine, that's going to stimulate hormone-sensitive lipase to release these fatty acids. And if we have insulin, that's going to kind of do the reverse. One way or another, once you get the fatty acid into cells, if you want to break it down for energy, well, that's going to happen in a process called your beta-oxidation. Beta-oxidation is going to break things down to acetyl-CoA at a time, and that's going to happen in your mitochondrial matrix. So we built up two at a time in our fatty acid synthesis. We could link those together to form triacylglycerols by linking them onto a glycerol backbone. And we could also break down the fatty acids through beta-oxidation, which was happening in our mitochondria. We wouldn't want to do both at the same time. And so that gives us into our idea of our reciprocal regulation. The key regulatory points are going to be our acetyl-CoA carboxylase when it comes to making fatty acids, and our CPT1 or our CAT1 when it comes to breaking down fatty acids. Remember that the CPT1, this CAT1, that was how we basically got these fatty acids. Well, we transformed them to carnitine, fatty acyl carnitines to sneak in. And so if we stop that process, we won't be able to get the fatty acids where they need to be in order to get broken down. And we can inhibit that when we have milanoil-CoA. Milanoil-CoA is made is an intermediate of making fatty acids. And so if we want to inhibit the breakdown of fatty acids when we make fatty acids, it makes sense that milanoil-CoA is used to inhibit this, especially since we don't really see milanoil-CoA in other pathways. So it's really kind of specific to the making of fatty acids. But if we have too much fatty acids, then those fatty acids build up and they feedback inhibit our acetyl-CoA carboxylase, which was making them milanoil-CoA, which we are using to make the fatty acids. So we get feedback inhibition. We also get feed-forward stimulation from citrate. Citrate was going to basically say, okay, you've got stuff coming, let's activate you. And that was going to activate the process further. Just like we had our hormone-sensitive lipase that was kind of going to be regulating the breakdown of the triacylglycerols into fatty acids. We also have hormonal regulation of our ACC. That hormonal regulation of our ACC was happening through phosphorylation of that ACC. Phosphorylation was happening in response to glucagon or epinephrine through our pKa. And then insulin was reversing things by acting a phosphatase that dephosphorylates things. This is a similar situation to the kind of reciprocal regulation that we saw when we talked about glycogen metabolism. If we have a lot of acetyl-CoA building up, well, this is going to cause problems. And so if we have a lot of acetyl-CoA built up, what can happen is we're actually going to get ketone body formation. These ketone body formation happens when the kind of like the last steps of your of the lipids breakdown of beta oxidation kind of go backwards because there's so much buildup. That makes acetyl-CoA, which can convert to make acetoacetate, which can make acetone, this byproduct, or it can be reduced to form beta hydroxybutyrate. So these are kind of like your ketone bodies that can travel through your bloodstream. Because they're carboxylic acids, they can acidify your bloodstream, causing ketoacidosis. And again, this can happen if acetyl-CoA builds up, which can happen if, say, you don't have enough oxaloacetate, such as in diabetes. If you're not able to kind of take in and break down sugars, then you're not going to have enough of this oxaloacetate to keep the cycle going. You're going to get CoA buildup. You can also get CoA buildup if you have problems with the kind of buildup of NADH, such as from alcoholism. If you have NADH buildup, that's going to inhibit your citric acid cycle. If you're inhibiting the citric acid cycle, the acetyl-CoA can't come in. The acetyl-CoA can't come in. You're going to build up acetyl-CoA. Some of it can be used to make fats, but if you outstrip kind of the speed at which you can make the fats, those acetyl-CoAs are just going to link up in order to make these ketone bodies. And so there are multiple ways in which you can have problems where you're generating a lot of ketone bodies, but the ketone bodies are also good when they're going to be used to fuel your brain.